Physiological interactions. Let's look at how plants defend themselves. There's a couple of different parts to this. This first part says constitutive or passive defenses. When we're looking at constitutive or passive defenses, we're talking about cuticle and wax layer, we're talking about Constitutive, we're talking about Sclerenchyma. These are a little bit tougher for the pathogen to get through. We need to deal with them. When we look at these, this shows you a corn leaf. On the outer part of the leaf, we have got this kind of rosé color material. That is your cuticle. Your cuticle and your wax layer are the first protective part. If the organism does not have an ability to penetrate through that, it will not get through. They have been able to demonstrate that there actually is a physical ability of things like fungi to be able to penetrate through this. When you get down into the other parts, you might be looking at Sclerenchyma or Constitutive, which again would be a hardier type material that is not as easily broken down. These create defenses. If we look at the outside of the potato, when you cut a potato, it will oxidize the outer part that has been cut. You will get a formation of what they call suberin, which is a cuticle-like material, which helps protect it from the outside. Potatoes like this have a thickened dermal layer. They've got suberin on the outside. It helps protect them from anything that might be trying to get in. Onion. The outer leaves of the onion have dried down. Since they are dried down, they are no longer living. You get this hardened material that serves as a defensive barrier. Green peppers. Outer part is dermal layer. It's got a thick outer waxy layer, and this helps protect it. They do spray these with wax on the outside to help it retain moisture as well. We have these vegetable waxes that are put on to help protect. They do work to a certain extent to keep pathogens out. We also have what we call induced or active defenses. These can be structural or they can be chemical. If we look at these two photographs, the one in the upper left-hand side, you can see where the arrow is pointing to a place where the pathogen has tried to put its penetration peg through. The plant is reacting to this by increasing the thickness of the cell wall at that spot. This is a response in there. So this is not a passive sort of thing. This is an active sort of thing. Whichever one wins this battle wins. If the plant can put down more cell wall such that the pathogen cannot ingress in, then the plant wins. If the pathogen can breach this area quicker than the plant can put up this part, then the pathogen wins. This particular part in here is called a papillae. If we look at the right-hand picture, it looks like a bunch of balloons. These are called Tyloses. As the plant determines that there is a pathogen on the inside, it will blow up cell membrane parts onto the inside of this to plug the inner part of the vascular system. When it does that, it limits the pathogen. Plant can also use gums. The plant can also use resins and materials like that in order to try and plug this up. If we look at the vascular system has these connections in between vascular filaments. Remember, the vascular filaments are not very long. They range from very short to somewhat longer in length. The size of the vascular tissue determines the length of the fibers. Wherever the fibers attach, there is a series of openings so that material can flow out of one vessel element into another. You can see them in here. They look like these chains in between these two cells. If you look at just the area, we'll see these little parts that we call pits. This is a gymnosperm type material where you can see the pits. If you look at an angiosperm, what you see is the pit ends kind of butt up against each other. We have what we call a sieve plate. That's what you see in here, the sieves that are the connection between the one cell and the next. The other cells are not near the sieve plate area. They just look like little open ducts. If you have a gum or something like that, it's going to plug up these connections. That is going to localize the pathogen within those particular parts. This is a rather unique thing. This is called a shot hole. As the pathogen gets in and it starts to move forward, starts to do things, the plant senses it, causes the tissue around the outside to die off, and the inner part of that lesion will fall out. Gives the appearance of what we call a shot hole. What's really happening is the plant is trying to keep the pathogen down. The way it does that is it puts these materials in there so it can quadrant it off. The material on the inside is now protected, or the rest of the plant is protected from the material on the inside. Then the material on the inside drops off. Over here we have the formation of resin. This is a material that is produced in response to some sort of attack on the plant that's trying to grab hold of whatever it is and tie it up so that it can't get out. This is fresh. You can see how this looks very clear. This is just starting to ooze out. It gets down like this and the volatile chemicals dry off. As it does that, you get the other material that is more oxidized. This is a peach that has done the same sort of thing. There is also what we call systemic induced resistance. This is kind of a new concept where they've looked at different types of plants. You can actually induce a response out of it by applying different types of materials in here. The question comes down to it, is this a usable sort of defense if we are looking at protecting plants in the field? And up to this point, we'd have to go no. It does exist. It's an interesting material there. When we look at plants, these are some of the ways that plants respond to try and win the battle against the pathogen.